you for having me today. It's really an honor to be here, and it's really exciting for me. This is the first time when I've presented on this seed color project publicly. We just had a preprint that came out um, just in like the last month, so pretty excited about this. So I wanted to start off by taking a step back and talking about the diversification of specifically harvested organs during the domestication process. And so no matter what crop you're working on, you probably are familiar with this huge range of variation that exists in the crop. Often it's not really commercialized, and it doesn't necessarily always fit in well with kind of a market chain in an industrialized country. But people have selected for this incredible amount of variation in almost every major crop, at least that people have been growing for a very long time, and have been really fully domesticated. So this might look familiar, and or if you're working with other crops, you're likely to know of similar patterns in your species. This is another example. These are all eggplants. Again, maybe you've seen things like this, but just the amount of color variation, shape variation, size variation is really incredible. And this is true of so many plants. And if you were to look at the wild relatives, often you see a very limited range of phenotypic variation. And so there's this contrast between the genetic diversity, which has declined in domestication, whereas the phenotypic diversity has really increased, especially in those organs that people are using. So in this case, it's fruit, sometimes it's seeds, like in common bean, but it also, you think about various root crops and all kinds of things, fruit, or I guess I already mentioned that, but there's so many different phenotypic variations. And this is something that Charles Darwin uh, attributed to potentially this interest in human novelty. And so I have this quote here from On the Origin of Species, and it mentions that it's in human nature to value any novelty, no matter how slight in one's own possession. And if you're the only one that has that, that white eggplant or the striped purple eggplant, whatever it might be, then if you're a subsistence farmer maybe, and you think that this is really unique and nobody else has ever had it, you're more likely to grow those things. And then there's this compounding pattern over thousands of years. So this is just a little bit of background, and I'm going to come back to this at the end a little bit. So we see this huge amount of variation in common bean, and also in fascioles generally. I mean, this is what I'm showing here is just a small proportion of the total variation in seed color and size and shape and all kinds of things. So why did we get interested in studying this? Well, we have on the one hand breeding projects that were focused on these like high value types, and California growers, especially like smaller scale organic growers, they want to have something that they can sell. Sometimes it's like at farmers markets or kind of local stores and restaurants and whatnot, but it has to have a unique market niche, because otherwise we can't compete with the lower production costs of the Midwest. So growers out here, they want something that's unique that they can charge a premium for. So we have breeding projects where we were specifically asked by some of our organic sector partners to develop varieties that had to, like, and we were working together to decide what varieties we wanted that really tasted good and had unique patterns and whatnot and would stand out in the market. So that was on the one hand. We're working with these for breeding projects. And then on the other hand, a lot of my research work has been on the genetics of domestication processes. You see there that GIF of shattering. And so that's the loss of pod shattering was an important part of the domestication syndrome in common bean and other legumes. And so that's one thing that we've looked at a lot. So we have a lot of longstanding interest in these types. We had them in our trials, and we were looking at them in detail. But really, this project began as I was looking at growth rate. I mentioned these organic partners. For them, weed competition is a really important characteristic. So they want something that grows fast and competes well against weeds. So we were growing out lots and lots of variations, seeing what grows the fastest. I wanted to draw your attention to this area specifically, where our fastest growing type, I'm just isolating the fastest and slowest. The fastest of these was black nightfall, you can see like days after planting and canopy cover, and the amount of canopy cover per plot of black nightfall is so much faster than this variety called Orca. I mean, it's really pretty astounding. So what do those two types look like? Well, here they are, black nightfall and Orca, and you can see that they have contrasting color patterns. And so I developed a population by hybridizing these and developing a few hundred recombinant inbred lines. And you can imagine that this then segregated not only for that growth rate, and that was the original intention of the population, but also for these color patterns. And so this is the kind of variation that we see in this black nightfall by Orca population. We also have the reciprocal cross with an equal number of lines. And so there's this huge amount of variation ranging all the way from black to white. And so the first thing that I wanted to cover in my talk today really is which genes were responsible. So I was really interested in this. Again, we already genotyped the population, we already have it. Now we can look into maybe what's going on in this population. So first, I went back to some of the old classic literature and saw there's Mark Bassett did all kinds of like traditional phenotypic screening and crossing of common beans for seed color, and he identified that there's this, it's called this half banner white, like it's this partial loss of function mutation, or he postulated partial loss of function gene that leads to this color pattern. I thought, well, that sure does look like what we see in black nightfall. So let's just say like maybe that's what's responsible there. And although he never linked it to this type. And then Orca is known to have Anasazi as a parent, and the genetics of the Anasazi seed color pattern have also been studied by Mark Bassett. And this is all, again, fairly traditional work. There are some linked rapid markers, but for these genes specifically, like that, yeah, T and BIP, there has been no gene model that has been proposed or anything, and really like the understanding of how these color patterns are achieved has not been characterized. So again, in Anasazi, the idea is that we have these two mutations here in T and BIP. Well, T is wild type, and then we have a mutation, that P gene, partial loss of function, whereas the other two genes are wild type in black nightfall. So then I went about making a genetic model of how to explain that range of variation that we see. And so I started filling this in, just a hypothesis to see like maybe this is what's going on. So Orca, we think has those two mutations. Black nightfall we think is there. And then how about, if you have dominant in all three, maybe you have a black seed, triple mutation, maybe you have a almost white seed, the lightest that we have. And then I kept filling this in. Some of this is characterized in literature. If you just have this, so this partial or this loss of function at T makes the seeds white, and that T is for totally colored seeds. So you have to have a mutation in that to have any kind of like real, like true white patching in the seed. So this is an important one, this T gene. Any kind of white patching, you have to have a mutation in it. But if you have that one alone, you'll have a white flower, but maybe just a little patch of white at the end. So it's really a combination of T and other genes coming together that's required for some of these like white patching color patterns. So this was our hypothesis. And we said, well, let's map this and see, basically put it to the test and find out where specifically these are mapping. So we did the QTL mapping, and you can see that we had these three major peaks, and when it's fully mutated, it leads to a white seed color, and that actually has been studied. And yet there are several partial loss of function mutations, and those have never been whatever would be responsible for them has not been identified. So we found that this was mapping to PT8, and I'll get back to that a little bit more. We found this gene called TTG1 on chromosome 9 that's maybe responsible for T, and then this third gene, there's this MYC1 that was mapping to BIP, and all very highly significant. So for this partial loss of function gene, again, we were thinking that maybe it's this transparent test at 8 ortholog, so TT8. That's an Arabidopsis gene. Again, this is only one of 4 known partial loss of function alleles at P, and the cDNA of those partial loss of function mutants was sequenced, but nobody was able to find any sequence differences. So there's something outside of the CDS that would be responsible, or it's a different gene entirely. So we thought that it was at least interesting and promising that it was mapping to that right location. And then for T, we found, and this had never been described before, but we found that it was mapping to this TTG1 gene. Maybe you see this here. I'm not sure if you can see my cursor, but this is a protein that's forming a complex actually with the last one I was talking about, at least in Arabidopsis. The 2 relatives are forming a complex together, and one's WDR, one's a basic helix. So it's 2 parts of these, what we call like these MBW complexes, or as my colleague calls them, BMW, which I like that kind of better. So it's easier to remember. So this is the B and the W of the BMW complex. So we thought that was pretty interesting. And then this is a close relative of the first gene that I was talking about. And so you see, this is an Arabidopsis paper, but you can see this is like a fundamental control of seed color in Arabidopsis. And what we're seeing is that 2 of our genes are very well like linked to this fundamental control in Arabidopsis. And so these are activating expression of some color, all the downstream color processes. And then I'll just say briefly, these complexes can form in many different ways. In Arabidopsis, there's always TTG1 that's forming the complexes, but then it can form with different bHLH proteins, and then those go on to form like different structures or govern different structures. And then there's even more of these MYB family transcription factors. And so TT8 is this one I've been talking about a lot. If you knock that out fully in common bean and other species, you'll get white seeds, and that's because that's required for all of these kinds of processes here. So then we're thinking that P is TT8. We're thinking that MYC1 is probably BIP, and T is the TTG1. That's our hypothesis. And again, these are forming clusters together to activate that expression. We also know that fairly recently, just in the last few years, some of the genes that are responsible, like the C gene is responsible for like a pinto spotting pattern that has been linked to MYB, and then there's this other gene that's also been linked to this 3rd part of the component. So it seems like what we're thinking is that maybe these 3 components have been mutated repeatedly, causing all kinds of color variation. So the next thing that we want to do, of course, is let's get some sequencing data. So we did whole genome shotgun sequencing on 37 types of diverse seed colors, and many of these we've used in breeding programs, and we know a little bit about the inheritance. Some of them are from our breeding program. We know a little bit about the genetics of the seed color behind them in many cases. And so we did this about 12x, just Illumina paired in 150 base pair reads. So just getting sequencing data across the whole genome and then aligning that and seeing where we have variation. And going back to this, I just wanted to highlight the first gene that I'll talk about again is this T gene. So again, it always leads to white flowers, at least in this Anasazi mutation, or Anasazi orphan mutation. And it also, as you can see, it has a lot of effects, but it is required for any kind of white patching on the seeds. And so we'll look at that in a little bit more detail. What we noticed immediately is that there's this 39 base pair deletion in Orca and Anasazi, and a lot of these types in our breeding program, these are Middle American. And so that 39 base pair deletion is going to be in frame. It's 13 amino acids that are clipped out. So that was pretty interesting. And I don't know if you guys are familiar with looking at this kind of diagram, but what we're seeing up here on the top, this is the gene model, and then this is showing where the reads are aligning. And so the fact that we have no reads that align to that part of the reference genome is an indication that that sequence is missing in the target. In this case, it's ORCA. And we even have reads that do span that. And then we have many other types where we that have this mutation, and they also have reads that span it. So then, again, 39 base pairs, in-frame mutation. But we had lots of types with partial white seed color, and what we saw was that there's another variety with a non-synonymous polymorphism, like SNP in a highly conserved residue, and that one has white seeds as well. We kept looking, we saw that there were 3 that have this 1 base pair deletion that causes a frame shift and premature stop codon, 1 base pair deletion in this other type in a different location, again, frame shift, premature stop codon. Several others had a 5 base pair deletion leading to premature stop codon. And then we noticed that some of our Andean types, these are from a different domestication event as the first one that I was talking about. So probably independently, all these things are probably independent, but this is a very different class that we're working with. And we saw that they have this really strange pattern in the UTR, the 3' UTR, where in all of them, there's some kind of a break going on. And looking at it in more detail, what we see is going on is that there's, based on where the mates are mapping, we can see that there's been this 14 megabase, this really huge inversion that cuts right there. And I don't fully know why that is leading to what seems to be a loss of function. But it is really interesting. And it's known that there can be regulatory elements in UTRs and whatnot, in 3' UTRs. And so it's certainly interesting. And what we know about this mutation as well is that it's the only one where you still have a colored flower, whereas all the other mutations will lead to white flower and seed patching. So it makes sense that there's some kind of different basis maybe to it. But again, maybe it's a hypothesis is that there's some kind of a seed-specific regulatory element downstream there, or alternate splicing that needs to be done, like for seeds, and something involved in that region. We found yet another mutation as well, where the entire gene is just completely gone. So here, I mean, you can just see there's just absolutely no reads aligning to this. The gene is right there. So we have this whole range of mutations, or what we think are mutations, all the way from single isolated SNP to several frame shifts to inversion to the whole gene being completely gone, and in-frame deletion. So we have this huge range of different mutations that have occurred, and in total there are 7 that we found just among these 22 lines. So it's clear that people have repeatedly selected this. And to me, this goes back to Darwin's original quote about like people being interested in novelty that's uniquely theirs. And clearly there have been many times in history when people have found some, interesting pattern, and specifically this like some kind of interesting white patch pattern in beans, and thought, well, this is interesting, I want to replant this. Certainly, I don't think there's any positive effect from these mutations that I'm aware of, other than just it's like a novelty to people. Okay. So then these are the protein, like the AlphaFold models of the proteins, and you can see that we have this whole range of premature stop codons and whatnot, and various deletions. Some of these, I'll talk about this one a little bit more later, but that ORCA deletion, again, it's just a clipping out of 13 amino acids. So it's not that different. And it's possible that this is affecting how it's interacting with its partners in those complexes. I'll get back to that a little bit more. So this is the second gene, and specifically it's regulating everything from the differences between the left and right here. And then what we found in that type is that there's a non-synonymous SNP this C217 position, and that is highly conserved. And it's known that that region is in the binding domain where it would normally interact with the last gene that I mentioned. There's no predicted major structural change due to this substitution, but if you look at the conservation of the site, it's extremely highly conserved. So 98 out of 100 of the most similar proteins have a C at that position, and and the two that don't are like predicted isoforms of a single soybean gene, but the main splice variant of that one has the C. So it's found everywhere in all these other related genes. It seems like, at least in some of their isoforms. I'm suspicious that the other thing is just a bioinformatic error. But again, so I mentioned 217, and so position 217 is in this, it's that WD40 like interacting domain. So it's interacting with the first gene that we were talking about. So again, forming a complex, and that mutation seems to be there in that region that would normally bind to the other one. And then we know in ORCA that there's just a small bit that's taken out, and this interaction on the other gene has many different like locations where it's touching. So basically, we're wondering whether or not some of these mutations might affect these interactions between the protein components. And then lastly, I wanted to talk about this P gene. Again, when it's fully mutated, we have a totally white seed type. We did include some white seeded snap beans that hadn't been sequenced here. We found that there's a novel deletion in this gene. So that's another like new total loss of function mutation in the gene that was common in all of the snap bean material. But then in the black nightfall types, for example, or this like hazy pattern type, what we saw is there's a deletion in an intron actually, and not in the exon, but it's this it's the second intron of the gene has a 600 base pair deletion, and we saw that in all of the types with that pattern. And it's very close, for whatever it's worth. It's very close to that intron. The gene itself is 14,000 base pairs, and this is within 18 of the exon. So here's another view of it. Up on the top, we see that the exons are this kind of tan color, and the black line is introns. And so this region here, black nightfall, this has been deleted, and this is just a multiple alignment of different species. And so red is showing like really strong similarity, and then the other colors are showing a lower degree of similarity. And so looking across legumes, some introns like in here just really have not been conserved at all. There's lots of differences, not a lot of alignment going on. If you look at exons, those tend to be pretty well conserved if you follow these down. Can you guys see my cursor? No.
Yeah, we can see your cursor.
Okay, good deal. Thank you. So the exons that are expressed tend to be more conserved, of course. But what we see in this region is that there are some sequence motifs that have been conserved going way up to some of these very distantly related legumes, and yet, so it's a suggestion that maybe it's doing something. If it's being conserved, it might have some functional impact, and then that's being deleted in black nightfall. Otherwise, black nightfall's like copy of this gene looks totally normal except for the deletion there. And then we saw that there's a deletion in a few other types. And if you look at this reference lima bean, that's what the seeds look like. So they are also partly colored. And then lastly, there's most cowpeas do have that sequence, but there's one type here. This is the best picture I could find of it. It's not very clear. Sorry about that, but it's also partly colored. And so then this is actually this is like a different legume subfamily, which probably should have just taken it out. But what we're seeing is that across all of this Papilionoideae, there's some conserved motifs, and then there are 3 types where there have been deletions, and all of them have partly colored seed coats. And so it's an indication that maybe this has some kind of functional impact. And indeed, there are papers talking about this and specifically saying that for a subset of genes, that the beginning of it, like early introns, are really important for gene expression. And so it's a possible explanation for why this has this effect. We then looked at the expression of the P gene, this important seed color gene, across our different phenotypic classes. And you can see that like this distinction between class 1 and class 4, the only difference is that intron mutation. And yet the expression of the this pigment gene is reduced by something like 20-fold just by having that intron deletion. And it's a highly significant difference. We also see, it's not quite significant, but we see like it looks like if you mutate T, there's certainly a major reduction in the expression of P. So you mutate one gene, and then the downstream target it has much lower average expression, and the two are not even close to overlapping. So seems like it's definitely some evidence for the effects of these, and specifically this intron deletion certainly seems to be very, very much involved. All of these types with the intron deletion have much lower expression than like a fully wild type line. And so those are all interesting results for us, but we also knew we have all this diversity, and we have whole genome sequencing data, and we might as well look at other regions of the genome that might be interesting for some of these other color genes, because we have the data sitting there. And so one of the other major mutations is a gene called Z, or like zonal, and it has been traditionally said that there are maybe 3 different alleles at this. So there's like wild type, there's a full mutation, then there's this Z cell, and this difference between Z, big Z, small z, was mapped to chromosome 3, and pretty close to an old scar marker. And then what we were seeing is that there was we were hypothesizing, is it possible maybe this is related to the MYB family transcription factors or something else? And if we look at these diagrams from Arabidopsis and how seed coat color patterning is controlled, you see that there's this TT2 gene that shows up all the time. That's transparent testa2, so like a transparent seed coat in Arabidopsis. And this TT2 gene, again, it seems to be very, very important. And we figured, well, it would be good to look at that in common B, and that's a MYB family transcription factor. And so, sure enough, we see that this PT2 gene equivalent in common bean is found on chromosome 3 there, and that's where Z is, and this is very, very close. So it's 1.4 megabase, so fairly close, and what we would expect based on the recombination distance. And if you look at the expression pattern of that gene, this is just from the Common Bean Gene Expression Atlas, it's massively expressed in seeds and basically nowhere else. So it's certainly very interesting based on its relationships with other genes known to influence seed color, the fact that it's expressed in the right location basically, and it's mapping to the right location. And so we looked at the materials at that locus, and we did see this like L105K mutation, so non-synonymous substitution, which was pretty common, especially in those white patched lines. And this is not predicted to be tolerated. And so we think that this very well could be responsible for that mutation. So again, K105, and I guess, and the 100 most similar proteins all have that. So it's clearly important functionally for the gene. And so by substituting it, you very likely have a reduction or loss in function of the gene. But interestingly, way back in the 90s, this difference between this like Salatus pattern versus like they're just these two different patterns. One is more like Salatus means saddle in Latin. One has this kind of it's almost like a hemispheric seed color pattern where it's in just like a smooth margin and whatnot, and the other one tends to be a lot more like splotchy. And we see more of what's on the far right, but we see some of these on the left. But that difference was actually mapped to chromosome 1, so a different chromosome. And in the literature since then, I don't think anyone's ever addressed why that is. And so we were thinking, well, maybe there's a 2-gene model here. And so maybe you have like one major gene that differentiates what's on the far left from the other two, and then there's a second gene that regulates the difference when Z is mutated. And we also didn't see, like between these two types, we saw the exact same mutation in that chromosome 3 gene. So there was nothing to explain the difference between them. So we looked into chromosome 1 and specifically found, and this had also been mapped quite close to 1 centimorgan of the determinacy gene. And we looked, and there's a very close relative of the T gene I was talking about, one of these MYB-bHLH-WD40 complex members that was basically right there. And we looked, and there's a deletion just almost like immediately right upstream of the gene in some types, and there's 4 in our panel with this mutation, whereas most of the other ones did not have a deletion, and there were lots of reads aligning to that region. And so if we look at what those 4 types phenotype might be, then here are those 4 with that deletion, and all of them have this kind of saddle-like, like kind of hemispheric seed color pattern with a pretty smooth margin, as opposed to like spots all over. And so I thought that was really interesting. And it's definitely a pretty strong candidate. With so many of these things, there's so much more, of course, that we would be able to do. So definitely like excited about the future possibilities. If we look at the expression of these 2 genes, here's Salatus and comparing it with Z. So the last, just looking at the expression of the last 2 genes that I was talking about, and we have this huge spike in expression in like seeds. So S, like common bean, Fasciculus vulgaris, seeds at the heart stage, seeds at 2 later stages. That's the only place that's really expressed, is right there in seeds. So that's another indication that's doing something specific to seeds. And it sure seems to, this deletion sure seems to be related to seed color. So we think that it's something exciting to explore. And again, even like having a 2-gene model for some of that had never been posed. So then the second and much shorter part of the talk is going to be on some of the environmental effects of this seed color patterning. And so we had some breeding materials planted out in 4 locations across California. And I don't know if you guys are familiar with California climate, but in the summer especially, the interior really gets very hot, and the coast stays very cool because of that like mild effect of the ocean. And so we had some sites that were like in view of the ocean, some that were kind of intermediate, and then we had these 2 that were pretty inland and quite hot. And we were growing out a lot of these materials with diverse seed colors, and it was really interesting. This is from the coastal site on the left versus Davis, which is in the interior on the right. And I'm much more used to seeing what's on the right, and that's what you've probably been seeing in this presentation most. So when we were out there then and harvesting in Pescadero, I was like, is this what's going on? And these were from the same seed lot. So we just took seed, just divided it into envelopes, planted at each location. And this is the kind of variation that we see in the harvested seeds. So that was really interesting. And then we look at across our types across all these different locations, and we're seeing it in many different types. So here again, Pescadero, the coolest site, stays very, very cool all summer. And then in Davis, it gets quite hot, and uh, there's this major difference in coloration across many different partly colored varieties. And then we can quantify that. So um, I made an ImageJ plugin to quantify the proportion of the seeds that's colored. And then we were looking at it across these locations. And sure enough, like the coolest locations on the left, in each case in that dark blue, warmest locations on the right in red, and the amount of seed coat pigmentation is always higher in the warmest location. And then we followed this up with a growth chamber experiment and looked at the look, and it was very well controlled. Of course, all the factors should be exactly the same, except that we're going to set the temperature different between the two chambers. And this is what they look like at a medium temperature, and then at high temperature, this is what the seeds look like. You can see that there's this major difference in seed pigmentation level, and high temperatures really do promote the expression of these phenolics across the pigmentation compounds across the seed coat. And here are the numbers for that. So again, like a 4 degree Celsius difference leads to often a 20% increase in or more in seed coat pigmentation. So definitely strong evidence that there is this effect of temperature on pigmentation. And then it's also worth pointing out that this Holstein mutation is different than has two genes that are mutated, both different, like independent mutations relative to these other ones. So we are seeing this in some different genetic backgrounds. So then, let's see. Then the question is, well, why do we see this kind of thing? And one hypothesis is that I mentioned that orca has two mutations that have to come together to have this kind of seed color pattern. And we know that in one of the proteins, that it's in that domain that interacts with the second one. And then in what we'll call the second gene, there was this deletion of a region, and maybe you put the two together and now they don't interact well, especially at lower temperatures, and maybe the interaction improves at higher temperatures. And so then the question is, if we are interested in making this more stable, and I think there is interest in doing quite a bit of interest in doing that, then would we be able to use other sources of these, like other mutations in the same genes to improve the stability? So maybe you remember me saying earlier in the presentation that we had one mutation where the whole gene is just gone, right? So if this is the result of differences in protein interactions, then if we tried substituting it with a mutation where the whole gene is just completely absent, it would be interesting to look into whether or not we still see this kind of difference in pigmentation based on temperature. Again, we know that this is happening in at least two completely different backgrounds. The other background is the one where there's a big inversion. And so we know it's happening in at least two different backgrounds, but I think it'd be worth exploring. And I do, in our conversations with industry and whatnot, we have been hearing maybe there's a need for this stability, right? You can't have something that's going to produce a totally different product from location to location, because a company that wants to sell, especially if we're talking about scaling it up, then we need it to be stable across locations. We can't have this kind of variation between environments. And maybe that's been limiting for some of these varieties to become commercialized in the past. So just in summary, I'm glad that we're finishing about on time. In summary, we mapped these three traditionally described genes to specific gene models that all form complexes together. And those did co-segregate completely. I didn't show everything that we did. We did more. We developed markers. We were looking at these mutations through the whole population, everything. And we found that many times these mutations were very highly parallel. So again, 7 independent mutations, even just in that T gene here in purple. And we also found additional mutations in some other genes that are also part of these MBW complexes. And those will definitely be things to look at in more detail in the future. And the one of the big ideas is that we think that these MBW complexes control this seed pigmentation expression across the seed coat. So even in our triple mutants, we get this little patch of color right there at the chalazial end of the seed, but it doesn't propagate out fully. Whereas when you have wild type genes, you get more and more expression across the seed coat. And so there's something about this propagation signal that is terminated in the mutants, but by raising the plants in warm temperatures, it seems to promote more expression of that color, and it promotes the propagation of that pigmentation activation pathway. And then there's plenty of future work that would be really exciting to me to keep diving into, looking at things like, can we improve the stability by using other existing mutations in these, and looking in more detail in some of the in plenty of things, the expression patterns of some of the other genes and whatnot. So those are some of those big ideas from the research side of things. And then I just want to conclude with some final thoughts. And looking at this, going back to this eggplant diversity, for example, if you look up these MBW complexes and their effects on lots of different things, I mean, you'll see that their role is really broad. I mean, it's not just even in like just in seeds, but they are really important for the activation of pigmentation pathways across many different organisms. So this is a dicot and this is a fruit, but then we could also look at maize, which is a monocot, and back to seeds here, more or less. And then again, MBW complexes. So it's very clear that these complexes and the genes that make them up have been mutated repeatedly, like each component. And to the best of my knowledge at this point, in common bean at least, every one of the mutations that leads to a pattern change in the seed coat, again, specifically pattern, every one of them that I know of is the result of a MBW complex member. Whereas we have other things where it's like the shade of color uniformly changing across the seed coat, and those will sometimes be enzymes or other things. But anyway, these MBW complexes clearly are very important. So I just wanted to finish up by saying thank you to my collaborators. It was definitely an international effort. We had people coming in from a few different people from Africa, and I'd like to thank my funding sources as well.
Awesome. Travis, thanks again for your talk today. Really appreciated it. Gosh, a lot of deep dive here, really digging into what's going on with the protein, the genetic look. Was really intrigued by seeing some of the inverted chromosome too that was going on there. I guess we'll open it up to see what kind of questions we might have from the group here. And as everyone's thinking on, go ahead.
Hey, I just wanted to say I really enjoyed your presentation. It was really, really lovely. I learned quite a bit. I just started working with dry beans, so this is a bit new for me. And I just really liked the concept that you also have that temperature affecting the expression of pigment. Just adds another layer of complexity to it, which makes sense when you look at how complex different seed colors can be in dry beans. So yeah, I just wanted to say I really liked your presentation.
Yeah, thank you.
Travis, I was kind of curious thinking about the T genes here. I know you kind of mentioned in the conversation, like there's just a huge variety of colors and options, and it seems like somewhat random selection from people based off of just personal preference of what they liked. Do you know, or have you seen in any like cultural instances where certain cultures might specifically be selecting colors or patterns moving forward? I mean, I think of like the white versus yellow maize, where certain cultures really prefer white maize versus
Mhm.
yellow. Do you see that in the bean patterns at all?
I I absolutely think that I mean, of course, there are major cultural patterns and preference. I mean,
Okay.
you just think, I mean, yeah, you would, well, and you would know as well as anybody, I think, about how much, you know, just different regions of the world. Like, you know, in Central America, maybe it's blacks and reds, and then in, you know, northern Mexico, it's pinto, and like you just look in Africa, it's calanquetes and yellows and these things, and
Hmm.
so there are differences. However, are there differences in like preferences for the specific white patching patterns? I would say maybe. I mean, and so much of this is like historical, and so I mean, I think that a lot of these, like the yellow eye pattern, seems to be popular in New England. Maybe we have some people
Hmm.
from New England here, but I definitely know of you know, there's like land races from New England there. Um, Paul, actually, I should have included a picture. Paul took a picture of like some landrace, like kind of, you know, heterogeneous landrace material from Bolivia with just such diversity of white patching patterns and all kinds of other patterns. And it's
Wow.
just like what you find in the market. Whereas I'm, I think that like these white, you know, white patching patterns are extremely rare from my experience in Africa, for example.
Hmm.
I'm sure they must have some, but I think it's very rare. So I would say that yes, um, there are differences in preferences, and so like this is that yellow eye type, that I think nobody would confuse that like with the Jacob's cattle, which Jacob's cattle are also popular New England heirlooms
Okay.
as well. So like this is just a very different pattern than this, and so I think
Yep.
that for people who are interested in these and yeah, interested in these kinds of things, they would never get them confused, and this is the result probably of this gene here. And
Interesting.
so I do think there's value. And if somebody was trying to sell like this as a yellow eye bean, I think that people who, I don't
Call
know,
your bluff on it. Yeah.
yeah, people know the difference. Yeah, at
Yep.
least yeah, as far as these things go.
Huh.
It looks like Henry raised his hand.
Yeah.
Yes, just as a quick question, thanks for the talk first. Have you guys observed anything related to the promoters of these genes, or have you ever thought of that, like could be related, especially in those experiments that or analysis that you did comparing different environments? Yeah.
Yeah, we're always looking at promoter sequences, and uh, for example, I mean, where is it in here? So this difference we think is, so again, this is like the difference there between these different color patterns. We think that's the result of this deletions just upstream of a gene. So it depends maybe on what your exact definition of a promoter is. I do think it's like a little over 100 base pairs. So I mean, maybe it'd be a little bit more of an enhancer, but regulatory sequence upstream of the gene there. Yeah. This is one example. We have we have others. We're all absolutely always looking at promoters, and I do think like very occasionally we'll have like some SNP that's, you know, upstream, and you kind of don't know what to do with it. I do like to like make a note of those kinds of things. I don't know if you caught, I think I left in right here when we were talking about this inversion. I just made a little note to myself. I probably should have just taken it out for this talk, but I'm glad, since you asked, I'm glad that I left it in. Um, I made a little note to myself that there's a SNP that they share 1,500 base pairs upstream of the transcription start site. I personally doubt it matters very much, but who knows? Maybe that is an enhancer element, and maybe the inversion is a coincidence, and it's just like a, you know, it's just like dragged along. Maybe that's the sequence, but yeah, we'd have to do a deeper dive into some of these things, and do more fine mapping and stuff. There's obviously tons of variation to dig into, and lots of directions to go with all this stuff. Did that answer your question?
Yes, great, thank you.
And then I mentioned also like there was that intron deletion, and then we were looking, since that was a gene that we were really focusing on, and then we did look at the expression. We saw major differences in expression because of that.
Cool, thanks.
Fantastic. If we don't have any other questions, Travis, thanks again for your time and for sharing your knowledge and wisdom here. We really appreciate it. Thanks again for today.